Okay, so let me change this. So can you guys see my screen at all?
Yes, I can see it now.
All right, all right. Okay, so let's just get right to um what we came here to do today, basically. And this is ACIT lab, like I said, and we're a research lab that focuses on bridging the gap or intersecting computational tools, artificial intelligence, and drug discovery. And this is our first webinar. This is webinar 1.0, and I hope and I'm hoping that in this webinar we would gain a lot and leave this webinar being better than we were before. So let's get right into it. So this is the outline for today's webinar, and looking at the introduction to drug discovery, traditional drug discovery pipeline. What is CADD? That's a computer aided drug discovery. AI slash ML in drug discovery. AI is the rave now. Everybody wants to do AI. But what does it do in drug discovery? What part does it play in drug discovery? And we're looking at the computer aided pipeline workflow. How does it work now that we are implementing computational tools? How does the drug discovery pipeline? Uh, how does it look like? What are the things that we do in a computer aided drug design pipeline? And we're looking at the traditional versus computer aided pipeline, looking at case studies, um, computational tools for computer aided drug design, challenges and future directions, conclusion, and the details of CAD Bootcamp 1.0, which actually a gift for everyone that attends this webinar. Okay, so let's get right into it. So, introduction to drug discovery. So, what is drug discovery? It's the process of identifying and developing new therapeutic compounds. It's a multi-stage process, spanning from 10 to 15 years, from initial discovery to market approval. It's a long process. This looking for the drug, testing the drug on animals, and you pick a drug, you pick a particular compound, you find out that, oh, this compound can somewhat as it has some therapeutic advantages. So we want to look at it more scientifically. So we take it, we go on to test it on in vitro models, that means on cell lines and all that, and we say, okay, wow, um, it gave you some wonderful reactions. Gave you some things that you would want to see, favorable reactions. Yeah. Let's say for instance you want to see it inhibit or cause inhibit a particular protein, and you run it on the cell line. You see that, oh, it actually did inhibit this particular protein. So you move on to in vivo, and that's when you're working on a whole organism, live organisms. So when you now work on it on in vivo, you find out that, oh, this drug, this compound is toxic. Yes, it is healing this thing, is curing this particular condition, is inhibiting this protein, but for some reason this rat or this mice or this rabbit or guinea pig, for some reason, has neurological disorder after using the drug as a side effect or adverse effect. So that drug is not good, because yes, it works for that condition, but it's causing something, actually is an adverse effect that is very, very dangerous. You find out that maybe the rat starts staggering, started losing balance, or went blind, or the skin, there are lesions on the skin, or you find out there are some lesions on the liver or on the stomach or the GIT. That's not cool. So you go back to the drawing board again to go and pick another drug, and you run it up again. So it's a difficult search problem, basically. And if you're looking, like I said, if you're looking for a particular drug for a particular disease, let's say a pandemic causing virus like COVID-19 now, and we want to find a compound that will cure it. Now, one thing you should know about it is that the chemical space, mind you, not all chemicals are drug like. All drugs are chemicals, yeah, but not all chemicals are drugs, yeah. So now there are estimated 10 raised to the power of 63 drug-like molecules available. And mind you, we have only 10 raised to the power 24 grains of sand on Earth. we have more molecules, drug like, not even molecules, drug like molecules than there are grains of sand on Earth. So now imagine examining each grain of sand one by one. Imagine counting one grain of sand one by one. In fact, I don't think anybody will sit down and count every grain of salt from a cup of salt or something. Even a handful of salt, nobody will sit down and want to start counting how many grains of salt are there, talk less of grains of sand on the earth. And much more than that is the amount of drug like molecules. So that's the challenge in drug discovery. There's a lot of molecules to test, there's a lot of compounds to test. So, and not only that, we want our compound to bind to a biological target. We want it to bind to something. We want it to either stimulate something, activate something, or inhibit something. Either we want a particular process to be activated, or we want to inhibit a particular process by binding to a biological target. And we have different targets such as proteins, nucleic acid like DNA, RNA, lipids, or carbohydrates. So those things are the activities we want to modulate. So we want to bind to them to modulate them, either to activate or inhibit, to produce a particular medicinal effect or a physiological effect. So as you can see, drug discovery intersect between the chemical space and the biological space. So there's a chemical space where all the drugs are currently there, and we have the biological space where the drug has to bind to something. It has to have a target. Where is it going to? We have paracetamol. Paracetamol is an analgesic. It has where it's going to. It has its own target. Yeah. We have drugs like cyclosporine. We have tacrolimus. You have where they go to. We have drugs like omeprazole, which is a PPI inhibitor. It inhibits protein pump basically. Yeah. So we have all these drugs. They have where they are going to. They don't just go anywhere. We have many drugs like that, and they have their targets. Yeah. So we cannot just have drugs and not just know where it is going to. You cannot just be ingesting any drug. Just because it is a drug doesn't mean it works for anything. I mean, I think we know that too well. So this is a diagram just showing the chemical space. Molecule 1 can work for protein 2, can also work for protein 1, can also work for protein 10. You find out that there are some drugs that work for many things. You take for instance chlorpheniramine. That's what we call protein. People use it as a anti-allergic. And you also find out that it's also useful for somebody that has catarrh, or so you are trying to like reduce or suppress the catarrh. Or also you find out that it also works for if you are looking to get, if you want to sleep, is somewhat sedative. Yeah, somewhat sedative. If you want to sleep, if you use protein, you will definitely sleep. Yeah. So the reason why it is like that is because drugs can have some features that allows them to bind to this target, bind to this protein, bind to this protein, but it might bind to one more than the other. That's why these to have side effects. Because you find out that you use a drug and they tell you the side effect is fatigue, the side effect is diarrhea. Maybe it is actually going to the stomach to mess up with the ANS activity um in the gastrointestinal tract, yeah, and cause some relaxation of the intestines, allowing the person to just be stooling anyhow. So drugs are like that. They bind to different target, they bind to different target. So what we want to do is to get uh discover the target, know the target, and also get a drug like compound that fits best, and as well as a very very good safety profile, has a good pharmacokinetic profile, and also a safety profile. So traditional drug discovery pipeline, you move all the way from here. You see the disease related genomic. This is where they actually find out, okay, what gene is expressed that causes this disease. Okay, what protein is being produced as a result of this gene? Let's take for instance the DYRQ1A gene. It is a gene that is implicated in Down syndrome related cognitive impairment. You see, Down syndrome patients, they are not always that um, they might not be exactly as sharp as people as people that don't have Down syndrome. Yeah, that is not to say they cannot go on to do great things, but they are at a disadvantage basically. And what causes that cognitive impairment? Through genomic study, we found out that the DYRK1A gene um, was related to that. So that gene basically creates the DYRK1A protein. So they discovered that that protein is implicated in the cognitive impairment that you find in Down syndrome patients. So now we have a target. We have a target. That's the target identification area. So we target validation as well, and then we move to lead discovery. So now we have a target. So now we now have to now start testing every drug that we have. That's the traditional drug discovery pipeline. So it's exhaustive. It is expensive. Imagine, okay, in normal, even in university setting, maybe in your final year project, a project takes about um 4 to 8 weeks. Yeah, we will be testing those drugs on animals. First, you have to first do acute toxicity. Then you do then you start running the compound or the drug on the laboratory animal. Then you extract the organs. You take the blood sample. Then you take it to where they will do analysis, biochemical analysis, and all those things. Very, very expensive. It's expensive. Now imagine having to do that for 10 raised to the power of 63 compounds. It is exhaustive. It is too expensive. That's why it takes so long for new drugs to be commercially available. Now, it takes 1 to 2 years for lead discovery. Now you have to optimize what you have, because yes, you'll be able to find the drug that works well, but it does not have a good safety profile. Let's say the drug is carcinogenic. Now you have to do some optimization on the drug, on the molecule. Maybe you remove a chlorine atom and put something in melty group, or you add an amine group somewhere, and try to like play around the drug so as to make it better. And you do that for like 2 to 3 years. Then you move on to preclinical tests. That means after all the whatever, you take it back to the lab again to test on various animals, because it's not just one animal you will do it on. You work on in vitro, you work on in vivo as well. And various laboratory animals have where their strengths, as regards whatever research that you want to do. Let's say for instance, rabbits. Rabbits is very, very good for dermatological tests. You need rats for GIT tests. you know and you move on to clinical trials afterwards. And clinical trials, 4 to 6 years. And clinical trials doesn't mean that FDA would approve the drug, and if FDA does not approve the drug, you are back again. So it takes a very, very long time. So that's why you find out that 90% of drug candidates fail during development. 90% of them fail during development. You can see $2.6 billion per approved drug. That's why some of those drugs are very expensive, because of the money that is put into the research, and the companies that did the research want to make their money back. So um traditional drug discovery, basically, it is expensive, it is high risk, and the methodologies are not just okay to solve the problems that we have at hand. So that's where computer aided drug design comes in. So what is computer aided drug design? So it's a multidisciplinary field that attracts researchers from information technology, medicine, pharmacology, pharmacy, medicinal chemistry, and major life science fields. Basically almost everywhere in under life sciences. That's every field that you can find under physics, chemistry, and biology. It's an intersection across all of them. Even mathematics also, that makes this field tick. Yeah. So it says it represents a fundamental convergence of biology, chemistry, and computational science, leveraging information technology to assist in the drug discovery process. So that the computational method that helps us to identify, helps us to design, helps us to select, helps us to optimize promising drug candidates on computer before they are then synthesized and tested in the lab. So that's what computer aided drug discovery is. So the goal is to expedite drug discovery. The goal is to refine drug discovery, make it faster, make it less of a trial and error based process. Like I explained, the traditional or work pipeline. You have to test the drug. It doesn't work, you go back and pick another drug. You go back, you go and pick another drug. But this helps us test those drugs in bulk. It basically helps us test the drug in bulk, and you find out that you've removed so many years out of the drug design process, out of the drug design timeline. And artificial intelligence and machine learning in drug discovery. So what is artificial intelligence? So artificial intelligence is basically teaching computers to perform tasks that normally require intelligence. You see ChatGPT, those are the rave, that's the rave. Now you ask it, oh, what is pharmacology, and it gives you that. That's not exactly something an independent system would give you unless it is put there by a human being. So what AI is teaching computers to perform tasks that require human intelligence. So you find out that it's like it's like a working person, like a brain, that recognizes pattern, makes decisions, solves problems, and even learns from experience. You find out that the more you use ChatGPT, the more it get better at answering the question the way you would like. Have you I believe every single person here has noticed that, you know, you find out that it's just, you know, it just answers question the way you would like it. It has discovered, it learns patterns, it recognizes patterns. And machine learning is just a subset of artificial intelligence. So it's where, you know, we feed computer with a lot of examples of something and let it discover patterns of its own. So like say, let's take for instance now, in a baby, you show it different pictures of dogs and cats. You tell it this is a dog, this is a cat. Show it another picture, you point out, you label it that this is a dog, oh, this is a cat, this is a cat, this is a dog, and everything. So when the dog sees when the baby sees a dog, after that training process, it will be able to identify that dog because it has been trained on different examples. Just the way you're able to know colors. Somebody told you that this is red, this is yellow, this is green. So when you see it somewhere else, you're able to identify it. I don't even understand what I'm trying to say. So that's basically what it is. So the computer figures out the rules by itself. Say, okay, a cat has whiskers. Cats are generally smaller than dogs. I mean, domestic cats are generally smaller than dogs. Cats sometimes can have stripes. Dogs have bigger snouts, and it learns those little features of the pictures you've given to the baby. Yeah, and take it as more take it as you like. It is data basically, and that's what machine learning tries on. You feed it the data, it learns the data, it learns the different features in the data, and from there it devises a rule on identifying, okay, which is which. That's basically what machine learning is all about. So it has been trained on data and able to recognize patterns. So why is it powerful for drug discovery? You know, it is good in such a way that now, since AI machine learning can recognize patterns. So what if we train it on a set of drugs? We put in a data where we say, okay, this drug is toxic, is liver toxic. This drug is not liver toxic. So we have a data set of thousands of drugs that are liver toxic, and we have also inside that same data set thousands of drugs that are not liver toxic, and we feed it into that machine learning algorithm or that artificial intelligence algorithm. It will learn the patterns. It will learn, okay, what actually does make a drug, because from the different examples it can learn what makes up a liver toxic drug, what makes up a drug that is not liver toxic. So when I come to query the machine learning or the AI to ask it, oh, is this is so drug a is also drug liver toxic? Does it have liver toxicity? Would it cause liver toxicity, or will it cause drug induced liver injury? So from the data that has been fed to it, it will learn from it, and to find out that it will give you a very very accurate answer. So that's basically what AI does. So all these years of research that has been done, that is not to throw away the traditional approach, because the traditional approach where the basis was where the data that the AI is being trained on is gotten from. That's where it is gotten from. But now that we have something that can automate the process, we've gotten a large amount. I mean, there's still a need for a lot more data because we want to make our machine learning or AI computational tools much more accurate at predicting. Because you would not like when it predicts the wrong thing. It tells you, oh, this drug is fine, it's not toxic, the safety profile is nice, and you use it, and you find out that every single specimen, every single laboratory animal that you just give to it, like, it just keeps dying or keeps having a particular condition or not, which is not good, because we are dealing with life here, and we also do not want to just be wasting resources as regards laboratory animals. So it helps us refine this, and it can analyze millions of molecules overnight, something that would take decades for scientists to test. So that's what computer-aided drug discovery is. It reduces time, reduces money, it reduces cost, it reduces trial and error, it reduces the use of resources, it reduces or excessive use of resources, so to say. So what is the pipeline? What is the workflow of a CAD drug design? So there are two ways you can go about it. There's a virtual screening and it's also de novo design. Virtual screening and de novo design. So virtual screening is where we predict and prioritize compounds as drug candidates by simulating their interactions with a target receptor or predicting other properties relevant to drug discovery. So what are we saying here? So what we are saying here is, like I said, the example I gave in the previous section, I said you train it on, okay, let's say we have a list of sympathomimetics, that means drugs that elicit sympathetic activity. Yeah, they elicit sympathetic activity. That means they are likely to activate adrenergic receptors. Yeah, they are likely to activate adrenergic receptors. So we feed it into the ML system. We feed it into the system for it to learn and say, okay, this is a drug that is a sympathomimetic, and this is a drug that is not a sympathomimetic. So he learns from it and learns that, okay, these are the things that a sympathomimetic drug must have. These are the functional groups. These are the structures. Does it have an aromatic ring bounded to an amine group? And it learns all those different things, the different nuances that sympathomimetic drugs would have. And with that particular system, it can predict whether a drug that it has not been trained on, it has not seen that drug before, but because it learns patterns, so it will look at that drug and look at it and see, does the pattern fit, is it similar to what I have, what I've been trained on? And if it does, then it will accurately predict that yes, this drug will most likely be a sympathomimetic drug, or yes, this drug can, yes, activate alpha 1 receptors or can inhibit beta 2 receptors. That's just basically it. Yeah, that's just basically it. So it learns. So it looks at the chemical space, and that consists of the entire set of molecules. So it learns from the chemical space and learns that, okay, this drug can work for this particular stuff. But now de novo design. Now with our massive with the massive research that's been done on drugs, on compound, medicinal chemistry, pharmacology, pharmacy, biology, yeah, scientists have been able to discover some certain functional groups, some certain things that you should find in a drug that would inhibit, let's say, dopamine receptors, that would say, let's say, CCK receptors, or G protein coupled receptors. Yeah. So it learns those things. So we've been able to discover those functional groups. So now those group of functional group, we call them a functional space. So now let's say we have a model that can actually learn that, okay, these are the functional groups that helps in creating a drug that will do this particular activity, that will do this particular thing. Let's say that would help with pain. These are the functional groups that you find in a drug that would help with pain. So what will happen is that in this de novo design, it utilizes a model that will now generate so many molecules. It will combine the functional group in numerous ways. Maybe combine an aromatic group to an amine group, or it will combine an OH group to an alkyl group, and just it's just like, I don't know how to put it, like as if you're arranging Legos, those Lego blocks. So as if you're arranging Legos, you can do it any form you want to do it. You just add H here, no, remove this H, add an ethyl group here, add an amine group here, add an hydroxy group here, add an etoxy group here. That's what the model will do. So the model will just generate so many molecules, and making sure that the functional groups that you've trained it with, that okay, these are the functional groups that are found in um that are found in a drug that is to work for this particular stuff, that is to work for this particular ailment, or particular, that is to target this particular compound. So that is what that's de novo design. So it's as if you are inverting drug discovery. That means you already know the functional group. You just want to, play around with them, and let's see if we come up with a drug that would work for this, and come up with a drug that would give us a good safety profile. So with virtual screening, we are moving from a chemical space to a functional space. That means we are testing a chemical space to see, does it have this function. But de novo design is, we already know these functional groups have this function. Now we want to create a chemical space of drug-like molecules that we want to say that are novel, and we want to say they can actually work for this particular stuff. So that's just basically the two ways you can go about CAD. And how do you choose? How do you choose? Now you want to work on a particular stuff. Do you know the ligand structure? Now, when you get your your protein, your target, your biological target from a data bank, there are online resources that you could get your proteins from. So when you get those things from them, there is always a ligand attached to that protein. There's always a ligand attached to that protein. Like, that ligand is hypothesized that if you want to modulate this particular protein, any ligand that you want to use to modulate this kind of protein must look like must be somewhat similar to this ligand that is associated, that you got from the data bank, that is associated with that protein that you've gotten from the data bank. So there's always a ligand that is associated with the protein. So when you retrieve a protein from a protein data bank, there's always a ligand that is there. So now, there are some proteins that don't come with a co-crystallized ligand. There are some that don't come with those ligands. They are called co-crystallized ligand. So there are some that don't come with those ligands. So if it comes with the ligand, that means you know the ligand structure. Then you move on to virtual screening. That means you are checking if this our chemical space has drugs that are similar to this ligand. So that's when you see you known target structure here. So before I move down there, so let's look at this place. It said no, you don't know the ligand structure. The protein did not come with a co-crystallized ligand. That means you don't have a ligand, but you know the target structure. You know the structure of the protein. You have the protein target, so you move on to what? De novo design, because you don't know what you are working with, but you know the structures. You know what could possibly create a drug that would work for this particular biological target, a protein, be it a protein or a DNA or nucleic acids in general. So in virtual screening, if you know the target, you move to structure-based design. That means you have the ligand structure, you also have the biological target. Yeah, you have the ligand structure and you have the biological target. Then you can use the structure-based design. That means, what do you mean by structure-based design? That means you can easily create a workflow where you can test if a particular ligand can bind to a biological target. If a particular ligand can bind to a particular biological target. But now, what if you don't know the biological target? So what you do is the ligand-based design. So what is ligand-based design? Now, we have so many drugs that work for malaria. Yeah, we have so many drugs that work for malaria, and let's assume that we don't know the structure that they are binding to, and we want to create another drug that works for malaria, because what we are looking at here is known target structure. That we don't know it. See the area. No, we don't know the biological target. So now let's say we want to create a drug for malaria, like I said, and we don't know the structure of what these numerous drugs bind to, be it artemisinin, be it chloroquine. We don't know what they are binding to. Let's assume we don't know what they are binding to. So we could create a model where we train this particular model on drugs that treat malaria, that have been confirmed to work for malaria, and we now bring our drug and use that model to predict whether it will also work for malaria. So to look at the features of all the drugs that work for malaria and note all those things, note all the different features, and compare it with the features of our compound that we want to predict whether it works for malaria or not. And if it doesn't match, it tells us no. If it matches, it tells us yes. So that's basically ligand-based design. So you are comparing ligand to ligand. But in structure-based design, you're comparing a ligand to a structure. That does it actually bind to this structure? Does it actually bind to this biological target? So that's basically what structure-based design is all about, and which is actually one of my favorites, as well as de novo design. But ligand-based design is not exactly as interesting. It's not interesting, but it's still fun. It's still a lot of pipeline, still a lot of workflow to do as regards that as well. So this is the integrated CADD process. So you acquire your data from public and private databases. You literature mining. You get your ligand, you get your protein. You do some computational screening, be it in docking, pharmacophore modeling, and ML prediction. Then you score your compounds, because you're going to be getting a large amount of compound that you want to run through the model, and you want to score them to check, okay, which one actually does have a good binding affinity score, because we want to work with a drug that is potent, because the higher the binding affinity, the higher the potency of the drug. Yeah. So the higher the affinity, the higher the potency of the drug. And let's say you're even discovering a drug that would bind to um let's say GABA receptors more than benzodiazepines. Yeah, you want to find a drug that binds to GABA receptor more than benzodiazepines. So what you will be looking for is a drug that would give you a higher binding affinity score than benzodiazepines. So if you are looking for it to be better, you cannot pick a drug that is lower than what is out there, because what is the use if it's not much more potent than what we already have? Do you get now? So that's the reason for scoring and for optimization as well. That means the reason for optimization is to reduce the side effect, reduce the adverse effect, reduce its possibly metabolic effect as well, and also look to improve its absorption, its distribution, and also aid its excretion out of the body. So that's where ADMET comes in, and pharmacokinetic profiling. So one of the first things, like I said, ligand protein retrieval and preparation. So there are different places you can get ligands. You can get ligand from PubChem. You can get it from Lotus. And PubChem, you get synthetic compounds, drug-like synthetic compound. You get it from PubChem. And Lotus database is for natural products, and it's compound that you wouldn't find in natural products. I mean, maybe from animals, from plants, from fungi. Those are chemicals from natural products. So if you want to deal with natural products, you come to Lotus. So you get your data or your library of compounds from Lotus. And if you want to get your protein structures, you go to the RCSB PDB. That's the protein data bank. It's a very, very massive library of proteins, and this is gotten from there. This is the human DYRQ1A protein. This is the structure of the protein, and this is the co-crystallized ligand there. This is the co-crystallized ligand. I don't know if you guys can see that. You find out that the protein comes with, as you can see, you see this tiny thing here. That is the co-crystallized ligand. So when you get it from your database, your protein database, it comes with a co-crystallized ligand. So what you will just be doing is to look to better the binding score of this co-crystallized ligand. And since you have a co-crystallized ligand, you have a structure, then you can look to do structure-based drug design. So let's move on. So the next thing that next one we're moving on is pharmacophore modeling. Now, what do we do in pharmacophore modeling? So we look at our co-crystallized ligand, and we try to note what are the features of this compound that is allowing it to bind to this protein. What are the features? Is it the hydrogen bond donors or acceptors? Is it the hydrophobic regions? Is it the aromatic rings? Is it the charged or ionizable groups, or the spatial arrangement of the compound? So that's where we know, okay, these are the features that each compound that would bind to this particular protein to modulate it should have. That means now if you look at this compound here, this is amine, binding to the DYRK1A protein, the one I showed you this page here. This is it here in the 3D space, and in one of the computational tools, this is Schrodinger mesh through. And as you can see here, it has 3 pharmacophoric features. That means the first one is an hydrogen bond acceptor A1, an aromatic ring R8, and another aromatic ring R7. So those are the 3 things that makes it. It has 2 aromatic rings that makes it possible for it to bind to this protein to elicit any activity at all. So what we'll be doing is that our model will be trained on this, looking for these 3 features in every compound that in every of our compounds in our library. So let's say we are planning to look in a library of 25,000 compounds, and we want to check, are these compounds are they likely compounds to bind to this particular protein. So once we've modeled it out to know where are the pharmacophoric features, so what we do is just look into those compounds, see, do you have these features? If you don't have these features, it moves along. Now there are different ways we can do it. We can say, oh, we want to pick, if you are working with very little compounds, basically you are working with, like, let's say 300 compounds, you can say, okay, our filter will be any compound that can have 2 out of 3 of these features would go to the next stage. Or you can make it much more strict and say it must have 3 out of 3 of these features. That means 3 out of all these, that means it must have this hydrogen bond acceptor, this aromatic ring, and this aromatic ring here. It must have it for it to move to the next stage. So by that, you've already reduced the compound libraries by 99%. This pharmacophoric modeling is very powerful. So you basically cut it down, because all compounds cannot bind to a particular protein. There is no protein that is like that all compounds just binds to it properly. And there's something we call a fitness score. So the we're looking for compounds that have these 3 stuff and have a fitness score of over 1, over 1.0. So basically, so that's basically what we're looking for. And you find out that it will reduce the amount of compounds that you're looking to test. Now, let's say you are coming with like 25,000 and 99% is gone. I mean, you are left with just 1% out of 25,000 compounds. You've greatly reduced it, and you would do this in matter of hours, rather than in an experimental pipeline where you have to test 25,000 molecules. Can you see the contrast between the timeline? Can you see the contrast between how long it takes for an experimental screening compared to a computer aided pipeline? So let's move to the next step, molecular docking. So after you've done your pharmacophore modeling, you move on to molecular docking, and there are so many tools that we use in molecular docking. You can use Autodock Viner or Schrodinger glide, Schrodinger glide rather. So this is where we predict the binding pulse. We predict the affinity of the ligand to bind with the protein binding site. Now we've gotten compounds that have those pharmacophoric features. Now we want to check the docking score. Can it actually dock? Now, drug and receptor, though there are theories that say it is not exactly like that, but we still love to look at it like that. That drug and a target, a receptor, a protein looks is like a lock and key. If a key doesn't fit into a lock well, it won't be able to open that lock. And it's how well it fits into that lock. That is how easy it will be to open that lock, basically. So that's the same thing for drug. So if it doesn't dock on the protein, the target properly, it won't elicit an activity that we would want it to elicit. It won't be as potent as we would want it to be. So like I said, we have our co-crystallized ligand, and we have our 1% that we've gotten from pharmacophore modeling. Now we want to dock them to see if we would see compounds that can dock well. Now, what's our aim here with this molecular docking? We want to see if we can get compounds that can have better binding score, can have better binding affinity than the co-crystallized ligand. That means the ligand that is associated with this protein that has been experimentally hypothesized that, okay, this particular ligand binds perfectly with this protein. And if we can get a compound that has a better binding score to this particular protein, that's a good candidate that we can move on to the next stage with. So when you do that, you've basically cut another percent. Out of that 1% again, you find out that you cut like maybe about 80% of that 1%. So you are left with a very, very little amount of compounds again that you take to the next stage as well. So there are series of docking on Schrodinger Glide, which is something a platform I'm very, very familiar with. There's HTVS, that's high throughput virtual screening. Then you have standard precision, there's extra precision. Yeah, and those three, yeah, as you go higher, the precision of the docking is much more better. And also as well, as you go higher, the computational demand is also higher, so it takes a longer time as you go to refine your process. So what you would do is that, let's say you start with HTVS, let's say you run 200 compounds through HTVS, you can say the top 50 compounds from HTVS will cross over to standard precision. So you run standard precision on those compounds, then you see the top 20 from there will move on to extra precision. Then from that extra precision, you can say the top 10 will move on to the next stage. And the next stage is MMGBSC, which is just like docking, but it is much more, it's not really docking, but it's a much more accurate way of calculating the binding affinity. It is very, very intensive. It is computationally intensive than docking. Docking is intensive, but it's much more computationally intensive. That's why you have to go through all those stages to reduce the amount of compounds, and you are stuck with only promising candidates. Only promising candidates. So imagine you having to run MMGBS of 25,000 compounds. It just won't work. It take a very, very long time. You need to have a large compute, which will use a lot of power, and to get those kind of compute, or let's say like a computer or a laptop that will do that, is quite expensive. And mind you, we're looking to make our process very, very cheap, cost effective as possible. That's the aim. Yeah. So that's the reason why you have to keep reducing and reducing as you go on. So the top 10 from extra precision moves on to MMGBSA, and the ones with the highest MMGBSA free energy score, let's say that top 10, you can still work with that top 10, because that top 10 is not a lot, so it can still run something on those top 10. So that top 10, so you rank them on their MMGBSA compared to your co-crystallized ligand. Mind you, we want to get better than our co-crystallized ligand. So, or it might not be a co-crystallized ligand, it might be a standard drug. Let's say, for instance, for GABA receptors, we know benzodiazepines are standard drugs for GABA receptors. So if we have a drug that has a better MMGBSA score, a better docking score than benzodiazepines, we have a very good promising candidate that we can move on to the next stage. And that's ADMET analysis. And we know ADMET, I think we have some pharmacology students here. And for the women are not pharmacology or pharmacy students here, ADMET is absorption, that's the A, distribution for the D, metabolism for M, E is excretion, and T for toxicity. Now, absorption, distribution, metabolism, and excretion are part of the pharmacokinetics. What is pharmacokinetics? Basically, what the body does to the drug. In as much as the drug does something to the human body, the body also does something to the drug. So first thing first, the body has to absorb the drug into the bloodstream. It has to distribute the drug to where it is needed, and it also has to metabolize the drug, because there are some drugs that are not exactly what works. There are some drugs that are pro-drugs. Let's say, for instance, for a Parkinson's patient, you administer levodopa. Levodopa is not what works in the body. The body converts it to dopamine, which actually eventually works in the body. So there are some drugs like that need to be metabolized and need to be converted to their active drug. And there are some drugs like that you are giving the active, the active one directly. You use the active drug directly, but because the body would always want to metabolize the drug so that it can be easily to excrete it, it reduces the amount of drug that gets to where it is needed. That means, let's say 80% of the drug managed to enter your bloodstream, and as it was going through the blood, goes into the liver and do some metabolic, because that's where most of the metabolism happens via the CYP450 enzymes. So if it goes there and it metabolizes, let's say about 40% away from it. That means you are left with another 40% out of the 80%. So, and let's say you need about 60 to 80% bioavailability. And when I mean bioavailability, that means the amount of drug that is inside the bloodstream that gets to where it is needed. That means gets to its actual therapeutic site. Yeah. So let's say it needs 60% bioavailability and you are left with 40%. The drug won't work. The drug just won't work. So if you have to look for a drug with a very good ADMET profile, and also toxicity as well. You don't want a drug that is hepatotoxic or mutagenic or carcinogenic or nephrotoxic or neurotoxic. You don't want any drug like that, because what's the point of using a drug when it causes another ailment for the person? So those are the things you have to look at. Now, out of your top 10, now you have your top 10, you can say, okay, these drugs are potent. Yeah, now out of these potent ones, which one actually has a good safety profile? Then you pick the top two from the MMGBAC analysis. You pick the top 10 from the MMGBAC analysis. You run your ADMET analysis on it, and you go on to do ADMET analysis. And you look at it, you compare it, and now your ADMET analysis depends on what you are working on. Assuming that, let's say we are working on, like for instance, I gave examples of the amine and human DYRK1A protein. You know, is a protein that is expressed in the brain, in the CNS. You know, it causes cognitive impairment in Down syndrome patients. Now, if the protein is in the brain, yeah, and you want it to get to the brain. So one of the things you will look at, aside from potency or docking score or MMGBSA score, is its BBB permeability. That means, is it permeable? Can it cross the blood-brain barrier? Because if you cannot cross the blood-brain barrier, it is not useful, because it's supposed to work in the brain. So it is not useful if we cannot cross the blood-brain barrier. Another thing again is liver toxicity. Those are the things you look at for. And also CYP450 inhibition or its interaction as well. You know, there are some drugs that inhibit CYP450 enzymes. Yeah, there are so many drugs that interact with CYP450. We have warfarin, we have ketoconazole. Those drugs, they interfere with CYP450. So you don't want a drug that interfere with CYP450, because it would affect any other drug that you would be using. Meaning you can't use any other drug as you are using that drug. Because let's say, for instance, now the drug is inhibiting CYP450, and you need CYP450 to metabolize the drug, another drug that you are taking at the same time as this drug. So you won't be able to take another drug. So those are a lot of nuances, a lot of things that you have to put into context while discovering a drug. And it has to go as a very, very good ADMET profile. Yeah, and there are so many ways to check it. You know, there's Lipinski rule of 5. So Lipinski rule of 5 is where you used to check if a compound is drug-like. But this one is even way at the beginning of when you retrieve your ligand and you do your ligand preparation. Yeah, and also there are many tools that we use for ADMET analysis, and we'll get there when we talk about the computational tools that we use. If you are following, can I see a reaction? I'd like to know if we are following. We are following this class. Just react. I don't know if I'm going too fast, if I'm going too slow, or if you understand what I'm trying to say. All right. Thank you very much. So, lead optimization. Now, we've picked our top two from ADMET analysis. We've said, okay, we've run ADMET analysis and we're picking our top two. These are our best two compounds. Now we have to optimize it. Yes, it has good ADMET profile, but we can make it better. We can actually tweak some things. We can add some things. We can remove some things that we actually do make it better. So when we are doing lead optimization, you can choose to do it manually. There's a software called ChemDraw. So you can choose to do it manually. So if you are an experienced medicinal chemist now, you would definitely know all those functional groups that would give you the features that you're looking for, like let's say activity or property that you're looking for. Let's say you want to make your drug more permeable to the blood-brain barrier. So there are some functional groups that you had that will improve that. Let's say, for instance, you had acute groups. Acute groups make your drug much more permeable to the blood-brain barrier. Also, we have fluorine, allergens in general. Allergens in general. Or let's say you're even looking to improve the docking score. You want to improve the potency. We have functional group like hydroxy, that's OH. And OH also helps with permeability as well. OH helps with permeability, but you do not want to do it too much with that OH, because the more OH you start adding, the more hydrogen bonds it will have. That means it becomes more hydrophilic and lipophobic. Now, once it gets lipophobic, and you know the blood-brain barrier, it is lipophilic, so the drug won't cross. And also, you don't want to do it too much with the fluorine and the chlorine, because these compounds have large molecular weight. So if we increase the molecular weight to some extent, it won't be able to cross the, even if it is lipophilic, it will still be able to cross the blood-brain uh, barrier. So those are a lot of things you put into consideration. So you could also run a generative model like the generative adversarial network, reinforcement learning, or diffusion models, where you just tell the model that, okay, I just want you to look at my scaffold. Like, for instance, look at this compound. While this was a project I worked on, and this was our lead compound, compound 25014. And this is it here. That gave us a very good ADMET profile and everything. But we still wanted to improve the ADMET profile. We wanted to improve the docking score as well. So you feed this into the model, and the model learns, okay, this is the backbone of of the drug you're looking to optimize, right? This is the backbone. Now we want to add or we want to remove some things from here. And as you can see, this is the analog that we created, the top analog that we finally add. And this is it here. As you can see, in this area here, there's another extra OH here. As you can see here, there's another OH here, there's another OH here as well. And we found out that it had a better docking score than this compound, had a better BBB score, had a better hepatotoxicity score. That means the hepatotoxicity was very low, the mutagenicity was very low, the carcinogenicity was very low. Now we've successfully optimized this using AI, using a machine learning algorithm to do that. And if you are looking to, unless maybe you are an experienced medicinal chemist, you know where to add, you know all the areas you want to add, avoiding those pharmacophoric features, selecting where exactly you want to add. If you're just a pharmacologist, or you're a pharmacist, or or let's say you are a science lab technician, or whatever field that is might not be exactly be versed in medicinal chemistry, yeah. So this model has been able to do this in matter of minutes to hours, when it might take you, because there are different combinations that you can come up with. Different combinations with this only, you can come up with about, there are different combinations, different ways you can arrange all these compounds, different ways you can add compounds. I mean, we've done math before, we've done combination and permutation. There's so many possibilities of combinations that you can have with respect to what functional group that you want to add. Let's say you want to add the halogens. There are like 4 or 5 that you want to add. Let's say you want to add OH group. Let's say you want to add a methyl group. Let's say you want to add so many things. Yeah. So many combinations that you can have. Yeah, and it is tasking. It is really, really tasking. By the time you do like 50, you will know that it is very rare. So that's what these generative models do for you. They make it work way much more faster. And with this generative model, I was able to generate 1,000 different analogs of this particular compound. And out of this 1,000, this was our best. Now imagine a human being sitting down to do 1,000 different molecules. It's not easy. I tried. In fact, I really did try, thinking I would do it, but it didn't work. I didn't get past 50. I didn't get past 50 myself. So, and we were able to get 1,000, and out of that 1,000, 438 were drug-like. Now, out of that 438 that were drug-like, that's the one we now took back into the pipeline again. So when you optimize, you have to take them through the process again and say, okay, we want to do pharmacophore modeling, want to do docking, want to do MMGBSA, and just like you did with your former library of compounds, you do it where you are cutting a particular percentage as you go on. Yeah. So, but now you're dealing with, like, let's say 438 compared to 25,000. So you do it way much more faster than 25,000 that you started with at the very beginning. So you see, you come up with this compound. This is molecule 358 out of the 1,000 analogs we created for compound 25014. So as you can see here, this is how 25014 docked to the protein. See, only this place had a binding. As you can see, this OHA bounded to this leucine here. But when we optimize it, you can see it is binding in different places. See, it's binding here, it is binding here, it is binding here, it is binding here, and all this gave us a way better binding score, and it in turn gave us a better ADMET profile as well. So you can see that changing functional groups, adding functional groups, yes, indeed, gives us a better or a different compound entirely, not just on the chemical formula, but in the properties, in its physiological activity as well. So comparing our original scaffold to our optimized lead, so the docking score was 13 compared to 10. And in docking score rankings, this is very high. Having 3 more than the original is very high. But on the MMGBSA score, which is very much accurate, though the 25014 had a better MMGBSA score, but in MMGBSA, if you are still under the 50 range, if you are still under like the tens range, let's say you're like 10 and 19, they're still very, very similar when we are dealing with MMGBSA. So though the original scaffold had a better MMGBSA score, but 57, 51 is still around the same range. It's not significant in terms of MMGBSA. But in terms of docking score, moving 3 steps away from the original scaffold is a lot, is a massive lot in terms of docking score. Now, BBB permeability, you're able to see that yet our 25014 was moderate, but we have that 48, our molecule 358, our optimized lead, had a better score. Yeah. As you can see here also, the hepatotoxicity here. This is human hepatotoxicity. It is a parameter in ADMET when you're doing ADMET analysis. So you see it here, the chances of it causing hepatotoxicity is about 56%. But here it's reduced. You see, we reduce liver toxicity risk, we reduce carcinogenicity as well. We also reduce the mutagenicity as well. As you can see, reduced all these toxicity that are very, very germane as regards our optimized compound. So let's move on. So virtual screening is much more faster. Basically, it's faster than experimental screening. CADD is very, very much more faster. Computer-aided drug design is faster, way much more faster. And AI coming to the flow, broadens, it helps us do things way much more faster. Yeah, helps us do things way much more faster. And de novo as well. In as much as we want to do virtual screening, yeah, virtual screening is also somewhat slow compared to de novo design, though they are all under computer aided drug design. But de novo is a little bit faster, but they have their disadvantages and advantages. De novo is faster. Virtual screening might be a little bit slower than de novo design. Now, the reason why virtual screening, the disadvantage I would say virtual screening would have is because virtual screening you find out that the drug would most likely have similar skeleton, similar structures to what we already have there. And because of that, it will be hard to patent the drug, to get a patent. Let's say, for instance, you want to create a drug that is better than benzodiazepine, but you're using benzodiazepine as your scaffold. Yeah. So it would have similar structure to benzodiazepine because you want it to bind to GABA receptor and all that. But you cannot easily patent it because the person that discovered the first the person that patented the very first benzodiazepine will always say, "Oh, you copied his work. You took some out of his thing." I know it's a very, very big thing in research. You do not want to copy someone. You don't want to steal someone's work without attributing properly. And you cannot just patent somebody's work as your own. You understand? So it's a very, very big thing as regards to this stuff. And also, it's something we call privilege structures. So these are structures that you frequently find in active compounds. Yeah. And the problem about privilege structure, why we call them privilege structures, is that they can bind to so many proteins. They can bind to so many proteins, like the benzodiazepines now. They can bind to so many proteins. And that is bad because you find out that benzodiazepine can bind to CCK receptor, can bind to HIV-1 reverse transcriptase, and can also bind to various G protein coupled receptors, which is not good because in as respect to this HIV, the benzodiazepine is improving the activity of the virus, is increasing what that virus is doing to the body. That's what benzodiazepine does in this regard. And that's just a side effect. That's something that it was not intended for. But because it has a privileged structure that it can bind to so many protein, it becomes harmful to the body. So that's where de novo comes in, because de novo will create a completely new compound that may or may not, but majority of the time, lack those privileged structures that might cause problem. But now the issue with the novel design, because you're coming up with a novel drug that nobody has seen before, that really does not have a basis, that we really don't have a basis for, yeah, is that it might not easily get approved by FDA. There will be more scrutiny in the clinical trials than when you use virtual screening. So it depends on what you're trying to achieve. If the novel design, achieves what you want to achieve, fine. If virtual screening does it, fine. Yeah. So you just have to understand the disadvantages and the advantage of these different methods and how you go about them. So let's look at traditional versus CAD pipeline. Now, another thing I wanted to also bring to you is that one thing about computer aided drug discovery. Let's say for instance you want to discover or you want to use a natural product. Let's say like for instance, there's a leaf called, I think the Yoruba people call it dongoyaro, or neem. That's the English name, NEEM plant, and it's found that if you use it to make a concoction or all those herbs and all those stuff, yeah, it cures malaria. And people looked into it that these herbalists have been using this thing for a very long time, and let's research this. And they researched and they found out that what was actually inside is quinine, the same quinine they found in cinchona bark. Yeah, it's quinine. It's the quinine content inside this particular plant that actually does cure malaria. Not that the plant is one miracle plant or something. They're able to find it. But now, before they were able to execute experiments on that particular plant, they had a basis. Now, when you want to take on a particular scientific research, you have to have a rationale. You cannot just pick things randomly and say, I just want to research on this. You have to have a reason why you are doing this. You have to have a rationale, a basis. What foundation are you laying your research on? So that's what CAD does for us, and it reduces the time taken to get a rationale. Because now, seeing that this drug has a good affinity, binding affinity, and a good ADMET analysis for this particular protein is a great basis to start from and say, it has been seen in studies that so and so has been good for this particular target, and that is why we are looking to translate that work into our animal studies. Let's say we want to use it on rats, on mice, on people. Some people do dogs, some people do chimpanzees and all that, guinea pigs and all those things. Yeah. So from there, once you are able to get a favorable result, then you will now use that in vivo result as a basis for the clinical trials. That means the in vivo result has shown it has been extensive research done in vivo and is moving on to clinical research. So you see, a stepping stone. But now, with the traditional pipeline, the issue with the traditional pipeline, let's say let's use that plant for instance. There are so many plants that they are of different species, but they have the same leaf. The leaf looks the same, and because they are of different species, that particular compound that actually treat that particular ailment might not be inside that other one. And you just find out that you just test and you just see that nothing came off it, and you just be like, what's going on? What's going on? So there's so many. That's just one of the disadvantage of the traditional way. Another disadvantage also is it takes time to identify a lead. It takes time to find basis, your rationale, and there's a lot of trial and error. There's a lot of money spent. There's a lot of money spent. But with CAD pipeline, with the CAD pipeline, it doesn't take a lot of time. Yes, let's say you even want to make your work as robust as possible. You want to make your work as elaborate as possible. Yes, 6 to 12 months it will take you to run a CAD pipeline. It depends on how robust you want to be. Depending on the resources that you have. Yeah, it depends on resources. And you can see $10 million investment before lead optimization and $2 million investment before lead optimization for the CAD pipeline. See the stack difference between both pipelines. And as you can see here, computer aided drug design takes, 1 to 2 years, 1 to 2 years. Let's say how many? 1, 2, 3, 4, 5. Let's say about 10, about 6, 7 to 10 years. That is going to be what reduced to 6 to 12 months by computer aided drug design, by bioinformatics, target druggability, library screening, docking, pharmacophore, and all these things will just be done in a matter of months, and something that would naturally take years, 6 to 10 years. Yeah, and I like to see the drug design pipeline like a funnel. See the traditional pipeline, the drug discovery. You see how large it is, clinical development, and FDA review, then to a drug. So as you can see, with drug discovery, yeah, this is where computer will come in, and as you can see, it is shorter. It is shorter and causing it to have less time taken to get a particular drug. So case studies, what are the drugs that have been gotten as regards computer AI or computer aided drug design? We have HIV protease inhibitors. We have saquinavir. We're able to get saquinavir. We're able to get imatinib, was also discovered with computer aided drug design techniques. And there are other ones as well. There's captopril. There's oseltamivir. There's boceprevir. And you see what they are used for. This captopril is an ACE inhibitor. I don't know if the pharmacology student or the physiology student or medical student on the call definitely know ACE inhibitors. And you see this HIV protease. This one works for HIV/AIDS. This one was a kinase inhibitor. This one works for neuraminidase and influenza. And this one works for hepatitis C and HCV protease. So these are examples of drugs. And if you want to locate, you go to this paper, this published paper, to see their workflow and how they discover this drug using computational tools. So computational tools that we use for CAD, what we use for visualization, basically, we have PyMo, OpenBabel, we have Schrodinger suite, we have PDB fixer. I personally use Schrodinger suite. Is one I'm very, very much familiar with. Schrodinger is paid, and the only way to get it is if you have an institution email and they have to approve that you can. They don't just allow anybody to buy the software or download the software or anything. They don't just allow anybody. So you have to use your institution. They have to approve you. They have to do a lot of checking and all that. So they don't just allow anybody. But I think there are other ones that are free. I think PyMol is free. OpenBabel is free, thereabouts. But for docking, if you want to run a docking analysis, a docking on, you can use Autodoc Viner, can use Glide on Schrodinger. Schrodinger suite is just a suite of applications. Everything that you need from the beginning to the end of a computer-aided drug discovery pipeline is in Schrodinger. It's a suite of applications or of platforms. So it's like Microsoft Office, where you have we have PowerPoint, you have Word, you have Excel, you have Outlook, you have so many things. Yeah, so that's how it is. Schrodinger. So Schrodinger is the one of Schrodinger has PyMol inside of it. It has Mistral. For binding affinity as well, you can use Amber or Gromax. You can use OcDoc. You can use MMPGBSA. Also, Schrodinger also has an MMGBSA calculation too as well. And for pharmacophore modeling, you can use FACE on Schrodinger. You can use Ligand Scout. You can use RDK Kit. You can use NIME Orange. I say NIME. This is their logo. And AI enabled platforms that you could use. There's reInvent, there's DeepDoc, there's Cynthia, there's DeepChem. You get deep learning models for molecular property prediction. There's MoleculeNet. This is where you can get data sets, models. AlphaFold also. AlphaFold was created by Google DeepMind. It was created by Google actually. Google created DeepMind, and yeah, so it's just a platform that can predict the structure of a protein. So because it's not a lot of it's not exactly a easy way to predict the structure of a protein. So we have a model that predicts almost near complete accuracy prediction of protein structures. And if we can have our protein structure, that's our biological target, it is easy for us to move on to our docking, pharmacophore modeling, and the rest. So integrated platform, like I said, commercial suites like Schrodinger. Schrodinger is a suite. And for ADMET analysis, you can use Swiss ADME or use ADMET Lab 3.0. I prefer ADMET Lab 3.0 personally. And data resources where you can get your compound libraries, where you can get your protein structures, you can get it from PubChem, and get one from Lotus. I think we discussed this earlier on. PDB where you can get your protein, Zinc database where you get drug-like molecules that you could work with, and yeah. So challenges and future directions, like you said, it needs to be trained on a lot of data, and data needs to be available, real data, um, data with correct labels. Because imagine as a child, your parents point you to your radio and tell you all your life, they'll be telling you that radio is a dog, and you see dog up and down, you just be like, that's not a dog. This, and you are pointing to a radio. Oh, see dog. That's messed up. Yeah, that's messed up. So I imagine that in the space. I mean that one is not, it might not necessarily be life threatening, but this one will be life threatening. Imagine a model misidentifying a compound as being drug like or being toxic or not toxic. If it misidentifies something as non-toxic, that means that drug is toxic and it could kill somebody. And if it misidentifies something as toxic, it means that drug could have been a perfect candidate for that particular ailment, but it has already misidentified it already. Yeah. So all these platforms already have all those machine learning models, AI models, deep learning models embedded into them already. So you might not necessarily have to code or type those code and all. Do me, I'm a much more technical person. I love coding as well, but I also miss, I think 50-50 personally for me. I mean 50-50 those platforms, 50-50 technical aspects, and yeah, but the introduction is just an introduction. Get yourself familiar with the computational tools that the models have already been loaded there, and it's much more easier. You don't have to have one proficiency, let's say in Python, in R or JAX, or one of those programming language that is used for machine learning and AI. But I mean, I'm proficient in Python, machine learning models, and all those things. But these are platforms that are industry grade. They are not just playful things or toy things. They are industry grade because they already have all those state of the art models embedded into them already, and you can make use of them as well. So like I said, the challenge is data. The data is also a very very big challenge, and also in as much as you want to do computer aided drug discovery, it does not remove you working on a living person or a living system. You have to test the drug on a living system because a living system is complicated, is very very sophisticated. On computer is maybe one or two complex But in a human being, human body is so complex, so sophisticated. So at the end of the day, you still have to test it. Does it actually work? Yeah. Even if yes, you've predicted that it works, that's just the basis for you to go on to do your clinical research or preclinical research, so to say. Yeah. So you can use that as a basis. So, but that doesn't mean CAD removes experimental validation. You have to validate it experimentally because the human body has so many nuances, so sophisticated, so many things working together to make the human body tick. And one drug can be the downfall of a person. So you have to test it. It's a must. Yeah. So the current limitations, like I said, quality depends on training data, garbage in, garbage out. So if you feed it wrong data, it give you nonsense as well. Yeah. So limited ability to model complex biological environments and the emerging solution as well. We're working on you know yeah, getting much more better data, much more better data that are correctly labeled, high quality data to make things much more better. So future outlook, what are we looking? Future directions, what are we looking for computer aided drug discovery to be like in future? We're looking for it to be autonomous, basically. We're looking for it to get very good, very very good, where we don't have to just press of a button and we've discovered like two drugs depending on even your kind of person. You're a black person, this drug is the best for you. You're a white person, this person is best for you. If you are an Asian, if you are a man or you're a woman, you're a child, you're elderly, you are pregnant or a convalescent or whoever, that's the aim, that's the goal, that's what we are trying to look for, to improve the quality of life consistently, constantly improve the quality of life. That's the goal, and we're looking for it to get as autonomous as possible. Yeah, before the advent of internet, a lot of people have to travel far to get their books. Now they can just get it on the internet, and now we don't have to even Google stuff anymore. I can't remember the last time I had to Google a particular information. I just go to ChatGPT to ask. Yeah, and that's uh much more autonomous, much more streamlined. And definitely, as we go on the next 5 to 10 years, you find out that there will be something else that would be much more better than what we currently have. So this also goes for every sector: drug discovery, health, medicine, finance, everywhere. You know, you find out that all this is what we're looking for. These are the future directions we're looking for in computer-aided drug discovery. So, conclusion: it significantly accelerates drug discovery while reducing costs, and as well it is transforming from supportive to central in drug discovery pipelines and rewards success validates computational approaches. I mean, we've seen about 5 examples of drugs that were a success using computational approach. So that for today's webinar. I said I have a gift. It's something at the end of this webinar, and I would like to share with every one of us the references for the presentation. And though it is in conjunction with NIPSA Unilag, that's the Nigerian Pharmacology Student Association, Unilag, and in collaboration with them, we've been able to come up with something, and in collaboration with AiSynth Labs, come up with a boot camp, a computer aided drug discovery boot camp, where get hands-on training, practical hands-on training on the tools, computer aided drug discovery tools. Learn it completely, from top to bottom, from the introduction, from getting the ligands, pharmacophore modeling, molecular docking, machine learning, AI tools that we use as regards this. You learn ADMET analysis, lead optimization with generative models, density functional theory. Let me expand this a little bit for everyone to see what we have. It's 6 weeks computer aided drug discovery bootcamp. It's just for 6 weeks, and get introduction, artificial intelligence, ligand protein retrieval, ligand based screening, pharmacophore modeling, molecular docking, MMGBSA. This is where we get into the nitty gritty of every single detail of what we discussed today, and it's going to be spanned across 6 weeks. And there's also going to be a capstone project that is going to be published on international journals. So if you're looking for to get a publication, and this is a valuable skill, I definitely know, I personally I know it's a valuable skill. It's helped me. And the valuable skill, and if you are looking to apply to grad school as well, to see a valuable skill to add to CV, to add to what you can do. You're not just a pharmacology student, you're not just a pharmacy student, you're not just a chemistry student or a medicine student. You are in tune, not just I know about this thing, but I know how to do these things. I know I have hands-on knowledge. I have hands-on practical knowledge of how this thing works, how the computer-aided drug discovery works. So, and this bootcamp is going to be tailored across so many research areas, be it in neuro, be it in respiratory system, cardiovascular system, antimicrobial resistance, cancer chemotherapy, HIV, COVID-19. So there's going to be a capstone project at the end of the day, which would work to get published as well. And you can have a publication under your belt, even, and this will be very very much perfect for fresh graduates, number one, and as well as people still in school. It's a flex. It's a flex. I wish I had it when I was an undergraduate. I had a publication under my belt. To be a massive flex, I'll be posting about it everywhere, and everybody just look at it. Because trust me, everybody looking at you at the top. Even without a publication, having this knowledge, you'll be seen as like a king or a queen that you are. And it puts you on top. It puts you way above your peers. Trust me, it puts you way above your peers, and you are confident in yourself. You're confident in what you have, basically. So that's what this boot camp is going to be all about. It's going to be for 6 weeks, and it's starting the 5th of January 2026. And there was an error with this thing. It's supposed to be 2026, but it's January 6th, 2020. January 5th, 2026, and the registration deadline is January 3rd. The registration deadline is going to be January 3rd. So do well to register for there's going to be a link after the slide. I'll send a link to the group as well. And you can also scan this QR code here as well. And it is for 15,000 naira. But due to our collaboration with NIPSA, it has been subsidized to 10,000 naira. So do well to register before the deadline, January 3rd. And I would love to see you around the boot camp and gain hands-on knowledge, practical knowledge on computational drug discovery, basically. I love to come around. And this is the link to register for the program, for the boot camp. And yeah, so I'll be taking questions now. If anyone has questions on what we've said so far, as regards the webinar, as regards the bootcamp, you can ask questions. Ask questions. We are open to questions right now. Open to questions right now. I'd love to hear your questions. So anybody have question? You have a question? Is there anything that you would like to gain clarity on? Yeah. Is there anything you would like to gain clarity on? Anything that you would want to know? Anything that you can just raise up your hand, signify, and ask questions. Yes, that's see me favor. Yes, the bootcamp is online. It's going to be on Google Meet, just like the webinar, and it's going to span for 6 weeks. Basically, it's going to span for 6 weeks, and it's going to be online. It's going to be online 6 weeks, and at the end of the 6 weeks, you've gained hands-on knowledge on computational drug discovery, and you are living with a project. You're living with a capstone project that is going to get published as well. And it is something that I believe, if one is very very intentional about, could also translate this into maybe you're in final year and you want to translate it into your final year project as well. You could look to want to synthesize the compound. You've gotten the compound and look to synthesize the compound. Try to procure the compound somewhere and look to work on the compound. Yeah, you procure the compound somewhere and look to use it to create something with it. Yeah, so it's something that is very, very beautiful, and I would love everyone to be a part of. There'll be more webinars, basically, more few webinars, basically, over next month. And okay, so need to have knowledge from Kofo. It says, do you need to have any knowledge skill prior to the bootcamp? No, no, no. There's really nothing. Just know how to use a computer. Yes, like I forgot to say, requirement is a working computer, basically. A working computer, preferably running Windows, running Windows, and you need to have a computer. Coding skills, no, you don't need to. I tried. The curriculum is packed with so much tools and approaches that requires no codes at all. No codes at all. So everything is already arranged and designed in such a way that you don't have to have a technical skill as regards coding. Just come as you are, and you know how to use a laptop, Windows preferably, like I said, and you are good to go. Yeah. So, does anyone have any other question? Any other question as regards the class? I want to answer more questions about the webinar. A laptop is fine. Yes, a laptop is fine. It's perfectly fine. I mean, I use my 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 Windows laptop because the apps are not exactly um available on Mac OS like it would be on Windows. So I use my laptop as well. So whatever machine you are using, a Windows machine, you can use it as well. So any other question? Any other question as regards the webinar, regards what was being said in the webinar, and you have any question? Any question? The webinar, the boot camp as well. I don't know if we understand what I said as regards in the webinar. Also, what is being taught in the webinar as regards computer-aided drug discovery. If I was clear, if you gained something from the meeting, please let me just react. Let's see what you've gained. If anybody that wants to share what they've gained from the meeting, just share. If you gained something from the meeting, just share. If there's anything that you've gained that you've never gained before, you can share. Yes, Koforola, go ahead.
Okay, good afternoon. So yeah, thank you so much for the webinar.